Connie began her PhD in marine biology in 2018. First, wanting to do population genetics, impact of invasive and non-native species. And she spent the first couple of years troubleshooting that. And then we had COVID, where nobody was really able to get in the lab and do any kind of lab work for a year and a half. And so after that, she decided that really to completely abandon kind of that line of study and to switch topics. And so to modeling benthic habitat, specifically macroalgae habitat. This has gone much better, where she has submitted one publication, one of her chapters, and the other one is ready to go. And the third one will probably be submitted sometime in the next couple of months. So as I've gotten to know Connie over the years, I've come to truly kind of appreciate her kindness and the valued role she's played within our team. Connie is always willing to lend a helping hand, whether it's diving, whether it's teaching her modeling techniques to the rest of our team, or answering questions about them, or identifying an incredible number of macroalgae either for our own project or for the Piscataqua Estuarine Partnership Program. What stands out really most is kind of her genuine curiosity about things, where Connie is never content with just knowing how, she really wants to know the why. And that not only in ecology, but certainly over the past few years, that's kind of transcended to algorithms and also how they're working as well. So her resilience and ability to pivot to a completely different topic within her PhD was very impressive. And so I just want to welcome Connie. And I also forgot to mention her her committee. Really everybody on her committee is really, I would say, been an integral part and team member of her thesis. And that is Chris Neethus, who's here in the room, Kim Lowell, who's here as well, Chris Parrish, and Dave Kucheski.
Thank you for the intro, Jen. Thanks to everyone who's here in person or online. I am going to be telling you about my doctoral research in habitat modeling in the Gulf of Maine. To first hint at why we're going to care about these kelp habitats that I'm modeling, I'm going to introduce the concept of a foundation species. Foundation species are a species or even a functional group of species that are numerically dominant within their ecosystem, non-trophically, through non-consumptive effects, alter the biodiversity of their ecosystem, and govern the flow of nutrient energy through their ecosystems. So for a terrestrial example, individual tree species are frequently considered, such as the eastern hemlock. We see a whole kind of mosaic of different tree species in the White Mountains here, and by dropping leaf litter versus not dropping leaf litter, decaying at different rates, having different nutrient concentrations, and providing different kinds of cover, they really govern what other organisms are living in and flourishing in these environments. In tropical waters, we have coral reefs. The reef-building coral species are often considered with that functional group kind of approach to it, where species with similar growth and calcification rates, similar primary production, those sorts of things are more grouped together in how they function. And in temperate marine environments, we have kelp forests as kind of traditional example. So we have a big macrocystis giant kelp forest. Here you see it's making a huge canopy, really reducing the amount of light coming through and creating a ton of this vertical structure. Foundation species often tend to be unidentified until they begin to decline now. It's hard to necessarily see the ways they're structuring environments until the function they're providing begins to be lost. There is an identified kind of worldwide trend of kelp decline and replacement of kelp habitats with red turf habitats. mapped here and associated with different stressors and drivers by Philby, Dexter, and Warnberg. So we see areas around the world where kelps are in decline and being replaced with kind of red macroalgae turf habitats with drivers such as warming, heat wave storms, pollution, eutrophication, and so on. This is what a turf habitat might look like around here. We have some healthy sugar kelp on the left side. This was at White Island, the Isles of Shoals, and some kind of red turf species off to the right side here. You will notice that these kelps we have are not the kind of tall forest-forming kelps that I showed in the previous slide, and we'll circle back around on that. But in the meantime, our area of interest here is the Gulf of Maine. That's the body of water extending from Cape Cod to the tip of Nova Scotia here, essentially. We're focused on the western Gulf of Maine here, the American portion of it, essentially, and with special attention paid here to this area off the New Hampshire coast here. So this star is Durham, where we are right now, if you're here in person, and this star is the Isles of Shoals, where a lot of my research is going to be focused, just a couple kilometers offshore here from Rye and Portsmouth. We have seen kelp decline occurring in the Gulf of Maine. This is a kind of decadal look at benthic habitat types at Star Island in the Isles of Shoals, and we see between 1997 and 2000 these kelps in decline, and the invasive green macroalga Codium frigio really taking over, followed by Bonomysonia homifera, a red turf species from 2000-2012, and then red turf species Desisiphonia japonica being very dominant in 2012 and 2015. So to show what kelp habitats in the Gulf of Maine would have traditionally looked like, we see here we have this short kelp canopy, these kelp beds with an understory of mostly native red macroalgae. So you see the way this kelp is kind of sitting a bit off the seabed here. It's creating shade, it's creating cover for a variety of coralline and other upright red algal species. Kelps are primary producers that physically structure the ecosystem by providing cover in this way and also allowing various predatory species to kind of partition space, have their own bits of habitat away from each other. Traditionally, the way kelp habitats were most often going to be disturbed was through these big urchin grazing fronts kind of cruising through and chowing down on everything and just leaving coralline crusts, which then through the process of succession, more species would reestablish until you kind of ended up at a kelp bed again. But we can look into some of the drivers that have caused conditions to move away from this kind of typical pattern over time. The key one is that the Gulf of Maine is warming rapidly. Pershing et al., 2015, we see that the Gulf of Maine is warming faster than about 99% of other areas of the ocean. With those rising temperatures comes marine heat waves. We've always had those, but they're getting more intense, more frequent, and they're lasting longer as well. This can cause lethal effects in some cases. Here in kind of southern New England and the Long Island Sound, we see declines in kelp following heat waves. And it can also have sublethal effects. So even kelps that survive heat waves, we see over kind of long period of time afterwards where they're not growing, photosynthesizing at the same rates they previously were. Warming also facilitates introduction and invasion of additional species because as the marine environment here becomes more similar in temperature to other regions of the world, as it kind of catches up since it's warming more quickly, species from other regions that are introduced here are more likely to be able to establish in favorable temperature conditions. Now here's what the change actually looks like in these habitats. So this is a video from White Island a couple of weeks ago. We see this kelp flowing nicely back and forth in the current. We see these cunner swimming around. They really prefer using kelp as habitat. When they're not foraging, it's a lot easier for them to hide in. It's much bigger, wavier, creates good habitat. These red turf species in the front, a lot smaller. So red turf species represent an alteration to that physical habitat structure and the way organisms living there use habitat. The initial invasion of Dasyphonia japonica reduced macroalgae diversity where it was growing at its most dense, and turf species like Dasyphonia also sheltered different assemblages of invertebrate species. Different kind of mesoinvertebrate small organisms that live along those branches are different than the ones that would associate with kelp blades. So that's something that's altering kind of the food web structure as well. So, but why do we care about these changes? Well, we can assign some dollar values to kelp, if that's helpful. Kelps in the Northwest Atlantic provide indirect fisheries value through habitat structuring and primary production, right? So species targeted by fisheries, such as lobster, crabs, pollock, make some use of kelp habitat. It helps them live at higher densities in some cases. And kelps also produce direct value by reducing carbon, nitrogen, phosphorus loading in water by using that and turning that into production instead. So we can assign a value of about $11 per square meter of kelp habitat per year. And that's not a lot of money, but there is a lot of habitat that potentially kelps could be living in. So it adds up. Kelps are also being increasingly targeted for mariculture efforts between Long Island and Maine. So because kelps are both ecologically important and provide value to commercial fisheries, it's important to understand the extent and stability of kelp habitats. So my research question is this: what factors determine the distribution and resilience of kelp habitats in the Gulf of Maine? And I'm going to use 3 objectives to kind of work through this research question here. First objective is to develop and assess a random forest approach for benthic habitat classification, and that's going to be focused on that Isles of Shoals area. Second objective is to use this developed random forest approach to identify patterns in benthic habitat and kelp species distribution and kind of dig into the variables that drive their distributions. And the third objective is to determine how effectively the commonly used MaxN approach to species distribution modeling can describe benthic habitats in the Gulf of Maine, because MaxN is a very frequently used model for species distribution modeling. We want to see how that relates to what we find from the random forest models. And that'll be for a broader Gulf of Maine extent. Now, to lay out how I'm going to dig into that first objective, I'm going to describe data gaps in long-term monitoring and kind of what we're not seeing, describe how modeling might improve our knowledge of these kelp habitats, and explain why I went with a random forest approach, and then describe the sources of data I'm using for modeling, the process of developing these models, and the steps to assess these models. So traditional long-term kelp monitoring that we do around here, that a lot of people do, tends to be limited in the amount of depth that people are actually going to. Of course, you can dive pretty deep, like Liz, but usually we're not. We are not really exceeding 12 to 14 meters most of the time out at shoals, because we have heavy equipment, we have to do a lot of diving in a short period of time, and we're not all expert divers, so we have to keep things kind of simple. But 14 meters depth only covers a pretty small portion of the area around the Isles of Shoals. So we don't necessarily really get a full picture of what's going on with macroalgae habitats below those depths. And this is very common. So here are a bunch of publications, many of which were used in that Phil B. Dexter paper to assess these worldwide trends of red turf habitats. And we see that most of them are also not exceeding 14 meters depth. The highlighted ones are the ones that are. But half of those are in Norway. So there's just really a lot of area where we're making these kind of broad declarations about how kelp habitats are doing based on only a small portion of their total depth range. But modeling has the potential to fill some of the gaps in kind of traditional long-term monitoring methods. We're looking at some bull kelp canopies in the Pacific Northwest here. They're tall kelps that extend all the way to the surface. We have the figure from Finger et al. Here again to see the side profile view. And because they grow at the surface, we can detect them at the surface using optical imagery from satellite or drones. Can use spectral indices to distinguish kind of the blades and bulbs of these kelps from the water around them. So, in the Pacific Ocean, where we have more of these kelp forests, it's possible to really just look down with optical imagery and see kind of what densities they're growing at, even though the kelp originate from the seafloor. On the other hand, our kelps don't extend to the surface. They don't get very close at all. And the ability of optical imagery to kind of penetrate to the bottom is limited. One approach was deployed by St. Pierre and Gagnon in the Gulf of St. Lawrence. So this is Anticosti Island here. They were able to pretty successfully model kelp habitats on the seabed using optical satellite imagery, but only to 7 meters depth. And that is still only a very small portion of that depth. That's less than we're typically diving to even. So while it is accurate and useful, it's still not something we can get the whole picture from. Acoustic approaches are less limited because we're not really worried about losing the sound waves. We can map very deep, as people in this building know. Porskamp et al. Here in Southern Australia used a acoustic bathymetry approach to modeling kelp and other benthic habitats. And while they found some success with it, that can't always be necessarily fully applied. It might not be appropriate here in the Gulf of Maine because what we see are red turfs and kelps preferring the same kinds of habitat in many cases. Red turfs are replacing kelp beds. So the sort of seafloor terrain variables we would describe with bathymetry are not going to be enough to get the full story here in terms of how these kind of habitat states are interacting with each other. Now I'll explain the random forest approach I'm using and then the data I'm going to use for it. Random forest models are non-parametric models that can be used for classification and regression. Non-parametric essentially means that they are making fewer assumptions at the outset, and they're pretty suitable for a wide range of sample sizes and complexities. They've been used for satellite-based marine habitat classification. We see some shallow tropical habitats in Indonesia here, mapped with random forest classification into a lot of different classes. They've also been used for bathymetry-based marine habitat classification, and random forests outperform a lot of other classification methods as well, like MLC, KNN, AdaBoost, CART, Quest. It performs similarly to support vector machines, but those are clunkier to use, so we favored random forests. Now, maybe by mentioning satellite and bathymetry data, I gave away the game a little bit, but now I'll talk about the data sources that I'm using in this modeling. We have Landsat 8 here, or a render of Landsat 8 anyway. This is an Earth-observing satellite that has thermal infrared imaging suite on board. We can take that thermal infrared data that it's looking at Earth and turn that into sea surface temperature data around the Isles of Shoals specifically. So what I did was take thermal infrared imagery and get SST data for July to September 2019 to 2023, and got median sea surface temperature and standard deviation of sea surface temperature during that period of time. The Landsat imagery is resolution-wise the limiting factor here, so it does limit the predictions of the model I'm building here to 30 meters resolution. We also have high-resolution bathymetry for a bunch of the Gulf of Maine, including around the Isles of Shoals. A lot of it sampled by the Hassler and by the Coastal Surveyor, which I don't have a picture of, so here's the Gulf Surveyor instead. From the high-resolution bathymetry, which we have at 4 meters resolution, we can calculate depth, slope, seafloor curvature, northness and eastness of slope, rugosity using Sappington et al.'s vector ruggedness measure, and we can use the thrust utility, which was developed in-house, to create geomorphon information too. So this is a map of geomorphons at shoals. These are essentially homogeneous terrain kernels. So the gray area are all areas that are considered flat. The green area are all areas that are considered slopes, and so on. Those were one hot encoded because random forests don't like unranked categorical data. So essentially turned into rasters of flat and non-flat, ridge and non-ridge, etc. Then we actually need information about where our habitat classes are occurring to predict on that. So affixed some GoPros to this camera frame here with a float, tossed it off a boat and hauled it back up again at a bunch of locations around shoals in the summer of 2022 and 2023, and turn those observations into a set of habitat classes. Ended up using 4, so bare substrate, we see a lot of sand dollars here. Kelp habitats, so this is a bed of a garum with a bunch of native red understory species. Mixed macroalgae habitat, where we have roughly equivalent amounts of red turf and kelp, and then red turf habitats. Next is the process of actually training a random forest model. So a random forest model is made of a lot of decision trees. So we take our habitat observations and the associated bathymetry and SST variables at the location of those observations, and then a decision tree, the classification tree, starts getting built off of a training subset of those samples. So random forest, anytime it's splitting data, it's offered a random subset of variables, and it's going to choose the one that lets it most accurately split up the observations it's working with. And then, next split, it's offered another set of variables, and so on until it's either run out of variables or classified all the data completely accurately. This is a simplified version. Usually there's multiple ways to get to any given habitat class that it'll kind of explore and be a little broader than this. But this is one of our trees in our random forest. All of the data that wasn't used to train it is used as validation data. It's run through the tree and classified. And since we know what the habitats are, we can get an error rate, and we can turn that error rate into an accuracy rate. So we grow a whole forest of decision trees. That's why it's a random forest. In a trained random forest, we have an overall accuracy rate from that process of looking at the validation data. We have variable importances in terms of mean decrease in accuracy. Essentially, for each of our variables, the random forest creates a permuted version where that variable isn't used and identifies how many more errors it would have made without it. Random forest can classify rasters with the same environmental variables, and they can classify data sets, and we can see how accurately they work with those. Random forests are not generally considered susceptible to overfitting, but a couple of researchers, Adelabu et al. And Barnyada et al., have found examples of it overfitting when presented with a separate test data set. So to mitigate any potential of this happening, perform Monte Carlo cross-validation. So essentially, split the data ahead of time into a training data set and a test data set. The training data set was used to build the forest and do that validation accuracy rate stuff as well. And then we split that habitat data a bunch of different ways up, always in the same proportions, but just breaking it up into different ways, and created n number of forests. So we can get the internal accuracy value from that training and validation subset, and then we can get an external accuracy value based on how it classifies that test subset. We can assess overfitting by subtracting the internal and external accuracy values from each other. The smaller the difference between them, the less likely there is to be any overfitting. And we can also get per class metrics, accuracy metrics using that test subset. So we'll see how each of the individual habitat classes perform as well. And then we can also use these random forests to classify habitat around the Isles of Shoals and see where it predicts habitats to be. But how do we set the number of simulations here? How do we pick how many Monte Carlo simulations? The Monte Carlo method, the more simulations you use, the more accurate it's going to be, but we hit a point of diminishing returns. But the more simulations we use, the more intensive that's going to be computationally, the longer it'll take. So we want to pick a good number where it's useful, but isn't taking too long. So ran it with different numbers of simulations, took a look at the external accuracy, saw where it started to stabilize, variance went down, and it wasn't increasing much anymore. and ended up using 175 simulations. There are also some hyperparameters we use to structure or constrain the random forest. So we have a forest. How many trees do we want in it? Basically, a very similar approach to the Monte Carlo simulations here. Test different numbers of trees, see where it starts to get stable. Pick a number that's not too high, but where we're not really seeing benefits anymore. So we used 215 trees. Then the final variable we're going to be concerned with is mtry, which governs the amount of variables that random forest is offered at each data. A tree is offered at each data split, whether it's a small number or whether it's picking between a lot of different variables to find the best one. We don't want to use too few variables because then we're going to have weaker decision trees in a random forest. But we also don't want to offer too many variables. Otherwise, these trees are all going to end up kind of coming out the same as each other. There's no randomness in our random forest in that case. So we want a nice intermediate value. So we used an mtry value of 6. Now let's take a look at how these trees actually bore out. So we had a mean internal accuracy of 71.3% and a mean external accuracy of 71.5%. There is very little discrepancy. I don't think we have any reason to be concerned about overfitting here. Then we can look at our per class accuracy metrics. We're going to take a look at a 1 versus rest basis for each class. Essentially, we can get precision, recall, and then an F1 score. So precision, essentially looking at everything in the circle here, everything the model classified as kelp, what proportion of it really is kelp. Then we can look at recall, which is everything that actually was kelp. How much of that did we accurately assess? And then the F1 score is the harmonic mean of precision and recall. We also have confusion data here. Just to take a look at this visually, we see true classes along the bottom. So essentially, bare substrate, accurately called bare substrate, kelp, accurately called kelp, and so on. We at least see that there's not really any effect where two classes are especially being confused for each other. They're all fairly proportional with each other. There's no real outsized effects where we're calling kelp bare substrate like half the time or anything like that. So we have higher F1 scores for bare substrate and kelp, which makes sense because they were also the more abundant observations, and slightly lower for mixed macroalgae and red turf. Those high F1 scores actually indicate that as far as kelp habitat classification goes, it's probably even a little more trustworthy than the 71% value, with mixed macroalgae and red turf being a bit lower, but still usable. But they were the less common classes, so there's some sense in that. Now, from our random forests, like I mentioned, we can classify rasters to get habitat predictions. This is the habitat predictions with depths below 50 meters mapped out. We don't have a hard limit on where particular, we don't have knowledge anyway of a hard limit of where particular habitats are dropping out at the Isles of Shoals. So 50 seemed like a pretty generous depth cutoff. So what we have here is we classified rasters with all 175 of those random forests. And since we have so many, we can do a threshold-based classification method. So we see that there are lighter and kind of darker beige in here. There's lighter and darker green. Essentially, those represent confidence levels. So we considered classifications where the same cell had the same classification in more than 75% of those simulations to be high confidence. If it was 50 to 75%, that was low confidence. And very few cells, less than 1%, did not have any majority class among those simulations at all. So we see generally these are pretty confident predictions. So we have 71% accuracy from these random forest models, which is pretty promising for just an initial modeling effort here. And we see higher performance with kelp identification in particular, based off that F1 score. And this does cover a greater depth range than existing monitoring. We dropped cameras down to 30 meters. So seeing this light green 30-meter depth limit, that's a pretty substantial increase in the amount of area, we can say more what's going on with the habitats in that area. Next, we'll take a deeper look at the random forest model outputs and see what information we can get out of those. So in our next objective, to use that random forest approach to identify patterns in benthic habitat and kelp species distribution, we are going to explore the variable importances from the random forest model. We are going to explore the predicted habitat class distributions, and we are going to explore the distributions of the kelp species as we observe them in the ground truth data. And the reason we're going to be interested in specific kelp species is because not all of the kelps might provide the same ecosystem services, and they're not all targeted for mariculture either. So there might be some differences going on in those habitats. So to start with the variable importances from the random forest, we have depth, vector ruggedness, standard deviation of median temperature, and slope as the 5 most important model-wide variables here. And depth in particular was the most important variable for both kelp and the red turf habitats. We have the flat and slope geomorphons and the other kind of bathymetric variables of curvature, northness, and eastness, making pretty solid contributions to identifying these classes as well. Ridge and foot slope geomorphons a little less slow, and then shoulder and valley not really doing much for our model here. When we look at these 5 most important ones and see how those really are helping us distinguish those classes, based on a Kruskal-Wallis test and Dunn's test, we see that depth is strongly separating bare substrate and kelp from mixed macroalgae and turf habitats. We see that areas with kelp are significantly more rugged on average than the other habitat types. We see that bare substrate has a significantly lower slope on average, which makes sense. Those are mostly sand flats, which flat is in the name. Not great habitat for macroalgae. We see that kelps are associated with the lowest median sea surface temperature, and that mixed macroalgae is associated with the lowest variation in sea surface temperature in terms of standard deviation. Now, to return to this habitat classification map, most of the area is bare substrate, lots of sand flats, like I said, but when we look at the predictions of the macroalgae habitat classes, we see that kelp is predicted to be 80% of that cover, with mixed macroalgae and red turf making up the remaining 20%. This is a lot more kelp than we expect to be out there, essentially. Looking at this graph again from earlier at Star Island, we see kelp kind of disappearing in the 21st century for the most part. But we should keep in mind that these data were collected at 8 meters depth. If we look at Star Island, because this was around Star Island, we see that there was a lot of mixed macroalgae habitat in kind of the waters near to the island, but as we move into deeper water, some kelp might be coming back as well. But we also see strong separation of depth here. So this black line here is a depth contour at 16 meters, and there are some pretty stark differences above and below 16 meters depth. 95% of the kelp habitat predictions are below 16 meters depth, and 99.5% of the red turf habitat predictions are shallower than 16 meters depth. But we also see that kelp species are being separated by depth. So in shallower water, we have more Saccharina latissima, sugar kelp. In deeper water, we have more Agarum clostratum, sieve kelp. These were also done with a Kruskal-Wallis test. These were also both statistically significant. While they both have pretty large ranges overall, Agarum does extend deeper and does occur most often significantly deeper than Saccharina latissima. And when we look at which is kind of the dominant species in a given observation, the kind of depth stratification between them becomes even more visible there. So we see a really strong depth donation of shallow water Saccharina to deeper water Agarum. We were not able to predict those kelp beds as separate kinds of habitat in the random forest model because we didn't have a ton of Saccharina latissima observations in there. But now I want to explore what role depth is playing in structuring habitats and structuring these kelp species here. Because we see shallow saccharina essentially competing with shallow red turf beds. But as we go deeper, there's not really dominant red turf anymore, and we just have these agarum beds. Light availability and temperature can both relate to depth, and those are probably things that contribute to the upper limit of agarum clethratum. It doesn't like areas that have too much light and are too warm. Saccharina latissima, its lower limit may be more set by light availability because it needs more light, but also by the ability of urchins to access those beds. Temperature and possibly light as well, colder temperatures at depth might contribute to the lower kind of depth limit of red turf dominance because Dasyphonia, based on Brandon O'Brien's thesis research, grow more slowly and reproduce more slowly in winter conditions. And agarum clethratum is resilient to urchin grazing, and that's probably something really important in structuring these species and structuring these habitats at depth. Because agarum is resilient to urchin grazing, kelp beds at depth that comprise agarum may survive for longer spans of time. And with all that space being occupied, there just might not be the ability for turfs, which might also be less competitive at those depths because of the coldness, to really set up in those areas and become dominant. For comparison, Whitman and Lamb identify healthy Saccharina latissima beds at Ammon Rock, and despite Dasyphonia japonica being out there, it's really not dominant like we see it in shallower water at shoals. But Whitman and Lamb also observe very few sea urchins. So if that's maybe one of the reasons that Saccharina is more stable out at that habitat, just there's more evidence in support of kelp bed resilience and stability being something that helps exclude turf habitats. So to return to the idea of kelps as foundation species here, I introduced kelp forests as a temperate marine foundation species. They really control the amount of light coming down. They really structure habitats. It makes sense to call these foundation species. But when we look at our kelp bed forming kelp species, these seem to tend to get really implicitly flattened into foundation species as well. When we're making global comparisons in kelp habitat decline, one kelp bed is kind of treated the same as the next kelp bed. But these have different functional traits and they structure the ecosystem in pretty different ways. So shallower Saccharina, they are doing more primary production. They're growing faster, they're growing larger. They are helping crabs and lobsters partition habitat, and they're sheltering understory species beneath them more so from wave energy and light. The deeper agarum beds, on the other hand, they grow more slowly. They're not getting as large overall, so they're less primary production there. They're resisting urchin herbivory, and they're sheltering understory species from urchin herbivory. And maybe because these are sheltered from warming and are better and are surviving for longer because they're not being eaten by urchins, they're just better at excluding invaders in that regard. But in any sense, I think it's important to consider these as really two distinctly different habitat states and not just collapse them into Gulf of Maine kelp beds. So to look at this Philby Dexter and Warrenberg map again, we're really making comparisons between all these kelp beds in these different regions. But we have 4 species of kelp in the Northwest Atlantic, more in parts of the Northeast Atlantic. Western Australia has a single species of kelp, Clonia radiata. So the diversity of species between region, or even diversity or changes in species with depth, can really change how resilient these environments are and how they're responding to things. So it might not always be the most appropriate thing to make broad characterizations globally about kelp habitats. But to kind of run through and circle back on our findings here in Objective 2, kelp habitats appear to be more resilient than traditional surveys are suggesting, but this resilience might favor agarum clethratum over Saccharina latissima, meaning we might see changes in ecosystem structure and ecosystem services as saccharina continues to be lost, right? Because we have the 16-meter depth contour, and it's really governing turf versus kelp, and that runs right through the area where saccharina occurs, and especially where saccharina is dominant. Those shallower areas are still very much affected. I've heard that saccharina mariculture is starting to focus on mid-water rather than kind of surface-grown kelp lines. If we can separate out these species and model them individually with future modeling efforts to get agarum beds and saccharina beds separately, that might help us identify areas where mariculture would be more successful, more preferable, and also areas where we can conserve and or restore saccharina in places where it could be sheltered better. So for our next objective, we'll zoom out to the rest of the Gulf of Maine and take a look at how we can apply the Maxent approach to species distribution modeling in the Gulf of Maine. I'm going to describe the use of Maxent and contrast it a bit with the random forests we just used. I'll describe the study area we're using here and some additional data sources I'm bringing in, some study species and the occurrence data sets we have for those species. Go through the process of cleaning and attempting to unbias those occurrence data sets, and then describe Maxent model development and assessment. So Maxent models are species distribution models that use only presence data. In comparison to a random forest model where we're comparing multiple habitat classes, Maxent models just need to know where something does occur. That's presence only data. Maxent models are frequently using existing occurrence data sets. Sometimes people are building them based on data they're growing and collecting themselves, but often they're using these existing resources, and that's what I'm going to show here too. Maxent can be used to forecast range shifts under future environmental conditions. So we have Wilson et al. 2019 here, mapping Laminaria digitata and forecasting it to some end-of-century climate projections. Maxent isn't always great at extrapolating outside of the sampled environmental space. So that might be where you'd want to use a what's called a hybrid model sometimes. You see Wilson, they've color-coded areas in red here and in yellow here versus green here, where while the model predicts laminaria in those areas, the physiological thresholds that are lethal might be exceeded. For our study area, we're expanding out to kind of the whole Gulf of Maine here. These are maps essentially of where we have bathymetry coverage from CCOM here. We have the western Gulf of Maine and southern New England on the left, which I'm just going to abbreviate as WGOM from here on. This is an area for which we have bathymetry initially gridded at 8 meters resolution, downsampled to 48 meters resolution, and further downsampled to 500 meters resolution. And I'll get into why I'm doing that. Then the smaller area on the right here, which you can see is a subset of the larger area, Plymouth to Cape Elizabeth, as well as Cash's Ledge. This is where we have high resolution bathymetry. That's going to be abbreviated as PTEAC. When I say it in my head, I say PTEAC, but I'm not going to say that out loud anymore. This bathymetry is gridded to 4 meters resolution. Downsampled it to 30 meters because of the Landsat data we're going to use, just like in the random forest model, and further downsample that to 48 meters resolution. So with this being at 48 meters resolution and with this downsampled, we can then make comparisons between these two kind of spatial extents. And we can also take a look at how some of these models change as they're additionally coarsened. Yes, there's some additional data here I want to include. Because Objective 2 showed the importance of light availability for individual kelp species, and since we're looking at species distribution models here, I wanted to bring in some data relating to light availability. So from Sentinel-2, I'm bringing in the normalized difference turbidity index and the normalized difference chlorophyll index, which use red and green spectral bands and red edge and red spectral bands to identify the amount of chlorophyll near the surface in seawater and the turbidity of seawater, which limits light availability. Sentinel-2 doesn't take thermal infrared imagery, but the bands used for these indices are at a finer starting resolution than the Landsat. Next is the occurrence data. So I'm using the GBIF data set. GBIF combines many species occurrence data sets from various institutions, so from museums, from university herbaria and collections, iNaturalist and other citizen science resources. And I'm going to look at 6 species here. 2 native kelp species, so Agarum and Saccharina, as we were talking about in the previous chapter. 2 invasive red turf species, so Bonomysonia and Dasysiphlonia. An invasive green macroalga, for contrast, Codium fragile. And then an introduced brown macroalga, Culpomania peregrina. Culpomania is an introduced species that's been around for a couple of years, but we haven't really established any concrete kind of nuisance or negative effects from it. So that would be an interesting point of contrast. To work with this occurrence data, I'm going to have to purge or fix any erroneous records that are in this data set. Going to have to correct for any spatial bias in sampling. And also, as we run the MaxEnt models, only use records with acceptable levels of coordinate uncertainty. We see, to start with, we have about 3,000 Saccharina records here. I just want you to keep that number in mind for later. I'll bring it up again. Dataset cleaning was an arduous process. I went into a lot of effort to do this. So for instance, I went through all the iNaturalist observations for all of the 6 species, had to remove observations where someone had brought something home and identified it, so it's georeferenced to their house. Can't use that. Any rack washed up on a beach, well, that's a valid observation. It tells us where that seaweed is dead, not where that seaweed is living. So can't use that either for the model. Also have this nice living kelp right in its, environment, but someone used the obscured geolocation tag, so we have 27 kilometers of coordinate uncertainty. So we really can't say where that is at all. There were also a lot of records where there was no coordinate uncertainty values, and since we can't assess how accurate or inaccurate those are, we can't use those either. There were also cases, especially with stuff from the UNH herbarium, where maybe the coordinates were fairly uncertain on the record we had online, but if there were site descriptions, I could associate those back with previous publications and find more detailed descriptions of where those were that the georeferencer didn't use when that was getting digitized, and improve some of those records. In other cases, I could do things like, for a certain Agarum occurrence, ask Chris Wells, who was at a desk next to me at the time, hey, where did you dive in Winter Harbor 10 years ago? And he was able to figure that out. So that was another way of improving record accuracy. After this whole data set cleaning process for these 6 species, I have to account for spatial bias, because there is spatial bias. At 30-meter resolution, these are the records we're left with for Dasysiphlonia japonica. That's really just focused on kind of New Hampshire here. Forcott et al. Suggests spatially stratifying your records, so essentially removing some records until the overall distribution is more kind of even. But we don't really have enough records to even do that with. We're just tossing them out and have nothing to replace them with. Moa et al. Suggest removing areas of environmental background known to be unsuitable to a given species. For instance, they said, okay, highly urbanized areas, Anopheles darlingi, they were doing mosquito modeling, can't live in those, so we can rule out any of those areas. We can't really do that the same way with marine species. Here is a rock washed up with like 5 Saccharina growing off of it. On a broad scale, if we're looking at habitat classes, like in random forest, we can say, okay, there's a sand flat, kelp's not going to be dominant there. But if we're looking at occurrence, if there's one good rock that a couple of macroalgae spores can settle on and grow, then it does occur there. So it's really hard to just categorically rule out areas and say there's not going to be anything here. Ended up using a method by Ellis to bias the environmental background to areas near records, essentially sort of a heat map here, where areas nearer records and the environmental conditions there are something that MaxEnt emphasizes more. I won't fully dig into this process, but essentially did distance accumulation raster from where those records were, converted that distance to probability using a Gaussian function with a 5 kilometer radius, because 5 kilometers is about the dispersal potential of kelp species. Weighted that probability using the number of cells within the radius, so nothing on the edge had a different emphasis, and then rescaled these values from 1 to 20. Maxent can take in these bias files and work with them directly. As for training and tuning the Maxent models, for each model, we want to start out by minimizing the delta AUC. To explain what that is, AUC is essentially the accuracy values for Maxent. So you have a training value and a test data value. And just like the random forest, the difference between those, the delta AUC, is an indicator of overfitting or lack of overfitting. If they're close, less likely to be overfit. Beta here is the regularization multiplier. That's essentially the amount of smoothing Maxent applies to the variables. Since some variables might be jagged, the amount of smoothing is a good thing to experiment with. Based on the initial model, we can identify the number of variables we can use at the end, because we don't want to have more variables than 1/10 the number of records Maxent is using. Once we have that number, we can perform stepwise removal of the weak variables, things that don't contribute to test gain or test AUC. And then we have our finalized models, which is predicted relative habitat suitability. From these models, we can compare the AUC between species or between spatial extents or between resolutions. We can compare overlap between species at the same resolution and extent using Schoner's D index. We can identify consistently important variables from the variable importances Maxent provides. And we can also take the models that we're building at 30 meters resolution, take a look at the Isles of Shoals, and see how the habitat suitability predictions compare to the random forest habitat class predictions. As far as Maxent model accuracy goes, we found that test AUCs were consistently high among models using Landsat, Sentinel-2, and bathymetry data in the 90 to 99% range, essentially. Asterisk here, I have something called BioOracle data. This was a very coarse, more kind of chemical oceanography focused data set that I tested out. I didn't talk about it more because it wasn't very useful, but you can see that the accuracy was lower, and it was also at a coarser resolution of 4 kilometers. It was 0.05 degrees per cell. So not super worth making comparisons with here, but I did it. While I don't report it on the slide, the delta AUC were also very minimal. There was rarely a difference in test and training of AUC of more than like 0.05 difference between them. Complexity in model development was frequently constrained by the number of records. We see at this 500 meter resolution here, the Saccharina model could only be built with 3 variables. But that's funny since we started with 3,000 Saccharina observations. 3,000 divided by 10 is 300. We didn't have 300 variables, but why would it be limited? We'll circle back around to that. Next is species overlap using Schoner's D index, so we can compare overlap between pairs of species at a given resolution and extent. The change in species overlap between resolution and extent is not predictable. So we see with Agaramendaza siphonia here, as we go to a more coarse resolution, the overlap between them decreases. Same with Agarament Saccharina, at least for that extent. Andesa siphonia and Saccharina also decrease in overlap, coarsening for the whole Gulf of Maine area. On the other hand, Agarament Saccharina for the broader Gulf of Maine area increase in overlap as the resolution gets coarser. And Dasasiphonia and Saccharina for the smaller extent also increase in overlap as the resolution gets coarser. So it's really, if there's no predictability there, it's hard to derive anything from these species overlap values. And that also means when you're comparing between different publications or between data you've collected, it's really important to know what scale you're looking at and how that might affect overlap between species if you're making those sorts of comparisons. Next, take a look at the comparisons between the Maxent and random forest predictions. So what we have here is a histogram of relative habitat suitability predicted by MaxScent, which is then chunked up by the habitat class predictions for those cells. We see at least that the areas predicted to be bare substrate by random forest are largely considered unsuitable habitat by MaxScent. So that's pretty good alignment there. But when we take that out and look at the macroalgae habitat types, we see it gets a lot worse. This is a garum, that's a kelp, right? Most of the area that is predicted by the random forest model to be kelp is considered unsuitable by garum, as is a lot of that area that's considered to be mixed macroalgae. In fact, the mixed macroalgae habitat category is frequently considered unsuitable by MaxScent, even though that should include all 4 of these species, which are kelps and red turf species. Next, briefly talk about the variable importances. Among the Landsat Sentinel and bathymetry models, depth, vector ruggedness, and standard deviation of sea surface temperature were consistently important, as they were in the random forest models. Median normalized difference turbidity index was important for Agarum clothratum and Dasisiphonia japonica, especially within the western Gulf of Maine kind of broader extent. But the chlorophyll index variables did not perform well and were frequently eliminated during that stepwise variable removal process. What this does bring up is the possibility that that NDTI data, since it was important for some of these, might work well if we go back and put that into a random forest models in the future. Also of note is that spatial bias, the bias file correction method really did not seem to work. We see where we had our observations of Dasisiphonia japonica, and we see the areas where most habitat suitability is predicted. We can't quantify the spatial bias because we don't have an idealized, unbiased data set to compare it to, but it really seems that the model is recapitulating the sampling effort and sampling bias, regardless of our attempts to fix that. The predictions are also inconsistent within a species, across scales and across spatial extents. So these are all Agarum models around the Isles of Shoals. Some of this, I think, plays into the different regularization multipliers because variable smoothing varied a lot and not in a really predictable way either. But we see that, for instance, at this 48 meter resolution model, for the smaller extent, there's a lot of this area to the west of the islands that's considered fairly suitable, where it's a lot more patchy and less suitable overall for the whole Gulf of Maine extent model in that area. And then much less of that area even is predicted to be suitable in the 30 meter resolution model. So the problem is that all of these models had high AUCs. They were all not overfit, they were all over 90%. So given that all 4 of these models are accurate, but are giving such different predictions, that makes it really difficult to, pick which one is more trustworthy than the others. I also want to point out that the coarser resolution models also lose a lot of area near the coastlines because of the window-based variable calculation steps. We see here at 48 meter resolution, you basically don't have any of the area between Appledore, Smutty Nose, and Star Island here, whereas more of that is retained at 30 meters resolution. And I think that plays into the loss of records as well. So coarser resolution causes a loss of coastal records because our sampling was biased towards near the shore anyway. More of that coastal area you lose, the more records you're going to lose. And coarser resolutions also create redundancy among records. So if at 30 meters, 2 observations were in adjacent cells, at 48 meters, they might be at the same cell, and MaxScent can only use one of those. And while at coarser resolution, you have more tolerance for coordinate uncertainty, that really wasn't enough to offset these. You pick up, 5 more Agarum observations, 5 more Bonomysonia, but it's not really enough to overcome the ways records are lost there. So we really see that the most records are used at the finest resolution model, which wasn't always enough to even build a meaningful model in the case of Codium and Copomania here. To go back to that GVIF data set that we were initially looking at that had almost 3,000 Saccharina latissima observations, At 30 meters resolution for part of the Gulf of Maine, we could use 125 of those. At 500-meter resolution for the whole Gulf of Maine, we had 25 records. So all of that cleaning, all that effort that went into cleaning gave us with very few observations that we could actually use for modeling. So to wrap things up here, we have stressors that are continuing to alter Gulf of Maine macroalgae habitats. But we see that remote sensing approaches that combine different data sources offer some pretty effective habitat modeling capabilities. In our first objective, the random forest models around the Isles of Shoals that we built had 71% accuracy and gave us insight into deeper areas that we were not really previously able to monitor at scale. In the second objective, we saw that both bathymetric and sea surface temperature variables were important in describing these macroalgae habitats. There was an important 16-meter depth threshold that marked the difference between where red turf macroalgae was occurring and where kelp habitats were dominant. And that depth is also approximately where we started to see a transition between kelp species as well. The random forest models suggest greater kelp bed resilience than we previously thought at the Isles of Shoals, but this seems to favor agarum over saccharina, which means that we're still probably going to see ecosystem and fishery services impact, and this affects how mariculture might be developed too. Kelps shouldn't be collapsed into a coherent, single foundation species. I don't think that's going to work because the functional traits are very different. We must address diversity and depth range, and we must try to model our kelp habitats as the individual separate habitat types that they are. With the MaxSet models, I found that existing occurrence data sets are very time-consuming to clean and for little reward. Spatial bias correction methods were difficult to apply to marine species compared to terrestrial species and don't always seem to be effective. They just don't necessarily work in correcting spatial bias. Despite the high AUC values, MaxSet models built on these occurrence data sets are very inconsistent across spatial scale and across resolutions. And it might be useful to at least we did find that it might be useful to incorporate turbidity data from Sentinel-2 or from other sources into these random forest models. Moving forward, I think the next logical step for the Isles of Shoals random forest models is to start expanding those towards the coast, towards Rye and Kittery, just sample closer to shore, see how the models generalize or see if they really need to be rebuilt or rethought as we expand the scope in that direction. Sampling needs to be adjusted so we can target each of the kelp species individually, because if we can model Saccharina latissima, and if we find areas where Saccharina latissima habitats are going to be more resilient, that does have implications for how we conserve or restore those areas and where we might want to set up mariculture. And if we can include turbidity data and incorporate that into these models, we might be able to see if that's helpful in identifying these habitats as well. I'll go into my acknowledgements and special thanks now. The NOAA grant that provided a lot of my funding. I had started with a teaching assistantship from the Department of Biological Sciences, but now I've been funded through a research assistantship by CCOM, and I've had some, that ill-fated population genetics study was also funded by a School of Marine Science and Ocean Engineering grant. I would like to thank my advisor, Jen, and all my committee members, Chris, Dave, Chris, and Kim. I would also like to give, there are a lot of mentors I've had over the years in undergrad, but I would especially like to give a shout out to Kyla Bennis. That was working with Kyla when she was still a grad student was my first time really working with seaweed. This is a picture from 12 years ago where I was calculating fucus surface areas in image J for her. I got to do more macroalgae work through the 3Cs program and by working with platform ocean acidification stuff at Northeastern as well. Did a kelp biomechanics research internship at the University of British Columbia. And just in 2021, I was able to co-mentor the Shoals intertidal interns with Kyla as well. So it was nice to go back and kind of be a peer there. I'd like to thank a lot of people from CCOM and from the Dykstra working group. So Kristen, of course, Brandon, Annie, Jordan, Caitlin, Gwenna, Melanie, Haley also for letting me gripe to you a lot, or like ask you questions when we're working next to each other, and Gabby for helping me with a lot of math stuff. I'd like to thank my parents, who supported me all along, and you know, if I didn't grow up practically at the beach, thanks to them, I might not be standing here doing this now. I'd like to thank some friends in particular, my girlfriend Abby, who made me like a week's worth of ZD while I was writing my dissertation. Really helped me get through that. And Jere A, who's hopefully watching right now, and a bunch of other friends, Sam, Chloe, Shannon, and Sarah. Any other friends and family who weren't mentioned, I of course also appreciate, but you know, I can't name anybody. And now I will take any questions.
You don't hold off, ask questions until the committee meeting after.
Liz.
Did you ground truth each of the different models?
So for the random forest, the way I collected the data initially was by doing those camera drops in those locations around shoals. So those were used for both model training and for kind of model assessment as kind of test data as well. The Maxent models were just really based on those occurrence data sets plus some initial observations, additional observations I added back in from dives we've done and stuff like that. Jenna first.
So fabulous job. Before you had any sort of insight based off of how depth influenced the like prevalence of different species, how did you go about building your sample data set? Was it kind of samples of opportunities? Were you doing both to?
I really just looked at a bathymetric map of shoals and picked areas that I thought would be different from each other. So just tried to cover a variety of, you know, this looks like a flat area. This is kind of on the edge of like a ledge over here. If I were doing it now, I would kind of take my data sets and do some like environmental space calculation and try to like cover that more thoroughly. But yeah, it was mostly just picking areas that seemed like they'd be different from each other.
Do you know what the depth distribution across your training samples were?
I don't have that on hand, but I sampled from, I would say like 6 to 30 meters about. Yes.
Great presentation. I thought it was so awesome. I really appreciated the tree graphics, and the way it kind of went from the yellow tree to like the very rectangular tree. Loved it. I also really loved your effortless pronunciation of all of the macroalgae that I can't even hope to. So love that. I also thought your this is going to be all compliments. Your presentation and the way that you've woven the story of how the data has these implications. It specifically for your application, I thought was amazing, effortless, fantastic. My actual question, I'm curious if you could talk a little bit about the temporal range of your two different, I'm going to call them separate studies, your random forest and your Maxent. I think you said your random forest was 2020 to 23, and the third one was like 1975 maybe to 22. I guess I'm just curious how you see this work fitting in with like monitoring methodologies, and how stable are these kelp beds? How much do they change?
Yes. So with the random forest, yeah, the ground truth data were collected between in 2022 and 2023 in the summertime, which is also why I biased the satellite temperature data to 2019 to 2023, just because the coverage is overall low, needed a bit of a wider range just to have a good amount of data in there. So yeah, that was, I really tried to get a kind of a snapshot there in the random forest model for the Isles of Shoals. For the Maxent model, that was something I wanted to do. I wanted to kind of take a more decadal approach to the extent that that was possible, because the high resolution temperature data doesn't go back very far, only to about 2013. But since there ended up just being so few usable samples, that kind of entered the equation. But by the point I was getting to that, it's like, well, there's nothing you can really do there. There's so few to build a model on to begin with that I can't really then separate that out by year. Thanks. Haley?
I have a question about like the sampling techniques themselves. So you use the drop camera for your data that's collected here.
Yes.
As you're like referencing literature, is that sort of the standard way to sample for kelp, or is that like what's preferred by you? Is it, do other articles use like diving or like other different approaches? Like, is there a standard way to survey, or is it just?
Diving is kind of the traditional approach still to kelp monitoring for the most part, it seems, and a lot of that is laying down transects or putting down quadrats at given intervals and stuff, which gives you a lot of nice, like, fine resolution detail on, you know, species diversity and stuff. But what the drop camera worked well with, I think, was kind of just doing those broader habitat types, and getting the wider amount of coverage from that. Was something that it's really hard to expand more traditional methods to. I have seen some stuff with video tows, but just didn't do those. Harder to keep track of where the camera is exactly versus dropping it, bringing it up without fancy geolocating equipment. Jenna.
So just because I'm biased towards robots, would an ROV help with getting a better geographical representation of kelp distribution? I know that
I'm certain it would, and it would cost a lot more money. Do we have any questions from the chat?
Nope, just a lot of congratulations.
Thanks. Okay.
It's a lot of scientific names. Any thoughts on the two other native kelps, Esculenta and
Alaria esculenta and Laminaria digitata. Didn't really observe those at shoals. They're not that common there to begin with, I don't think, but they just didn't show up in any of the camera observations, really.
the second part of that was just, are they rare to have shown up in your frontier surveys?
Yeah, shoals, absolutely. Yeah, I've seen some on the outside of Nubble too, I think, right? Like farther around.
So yeah, so we'll take a
Okay.
10-minute break and then our committee will kind of meeting here.